Our next speaker is Jonathan Cavanagh from the University of Glasgow. Thank you for making it here. I'm sure it's so rushed. And Jonathan will be talking about inflammatory aspects of mental health and psychiatric disorders such as depression and schizophrenia. Thank you.
Okay, thanks very much. Can you hear me? Yeah. It's a great honour to be here, back in Edinburgh after all these years, and thanks for asking me, BNA. I'm a rather strange psychiatrist. There's a lot of competition for that title, probably, but I work in both a wet lab environment and clinically. So I'm going to say a bit about both, if we have time. And what I work on is the neuroinflammatory mechanisms of really sickness behaviour. And sickness behaviour is a constellation of symptoms, really, that you see in all mammals once they're infected or inflamed. But they constitute three main areas. One is change in mood or reward or hedonic behaviour. Secondly, cognitive dysfunction. And thirdly, fatigue. As you can imagine, these are all things that are very relevant to most psychiatric phenotypes. But we have something of a problem, I think, mainly because I can't advance my slides, but there we are. The current problem, I think, can be summarized in three ways. One is that we don't really have any mechanistically specific biomarkers in psychiatry, none really worth talking about. There's a paucity of novel, tractable molecular targets. And part of that is driven by the lack of predictive animal models, in my view. So today I want to say something about mechanistic studies we use a variety of different animal models, but I'll pick two of them. Say something about the limitation of human studies to actually tell us about mechanistic pathways and therefore the importance of reverse translation. And then one example of an experimental medicine study that we're carrying out currently. So as I say, my group splits roughly into two. This kind of circular bit here is the clinical studies where we do a variety of different experimental medicine studies, and the rest is rodent-based. So why inflammation, or why is immunology important in any of this? Well, I think there are four main threads of argument, really. One is that inflammation itself, or inflammatory proteins, can induce depression, and I think that's perhaps the most powerful piece of evidence, in the sense that if you suffer from a disease that requires, say, interferon therapy, all right, so hepatitis C, some types of cancer, interferon alpha is typically used to treat those, or it certainly was. And that interferon is an inflammatory cytokine, okay? Now, if you give that to someone, about 50% of these people will become depressed de novo. They've never been depressed before, they develop depression, and intriguingly, that is maintained. They maintain the remitting, relapsing pattern of depression after the therapy with interferon. So once the therapy's ceased, once the disease underlying it is better, they continue to develop this remitting, relapsing depression. So there's some sort of priming going on here, which I think is biologically interesting. If you measure cytokines and chemokines in the peripheral blood of psychiatric patients, I mean depressed people, bipolar people, some people with schizophrenia, then you will see in a percentage of them, round about 20 to 25% of patients show an increased level of inflammatory biomarkers. Now, these are not particularly specific and not particularly high compared to disorders like rheumatoid arthritis or other conditions, but nonetheless they are greater than the healthy population. The other argument is that inflammation seems to be, as it were, coterminous with this treatment resistance we hear about in terms of antidepressant therapy. We heard from Beata about the benefits of antidepressant therapy, but still a stubborn third of the population, not that they're stubborn, but there's a stubbornness in their failure to respond. And that has been the case really since the advent of antidepressants. So to me, these are biologically different phenomena. And that resistance seems to be also, as I say, coterminous with this inflammatory profile that you see in those patients. And then the fourth is the converse of that, really, which is that anti-inflammatory therapies seem to provide some betterment in depression for some patients. And that's intriguing for my colleagues in things like rheumatology and neurology, where I work clinically. So I see inflamed patients who've got psychiatric phenotypes. So I spend most of my time with MS and Parkinson's and rheumatoid patients. And they almost all, without exception, exhibit those sickness behaviours I was telling you about. So mood change, cognitive change, fatigue, it's ubiquitous. And what's intriguing about the biological targeting in these disorders, which has revolutionised that field, actually. So when I was a medical student several years ago, rheumatoid arthritis patients died in their 50s, all right? They died of the condition, they died of inflammation, they died of renal failure because of the drugs we used. That was revolutionized by targeted biological therapies which target a specific cytokine. MS has been revolutionized. MS field was you know, a therapeutic desert up until relatively recently. So these new anti-inflammatory therapies have really changed medicine almost beyond recognition for some conditions, but not yet psychiatry. Let me just go back a bit. Those just that on your right-hand side, those are some meta-analytic studies showing the benefits of these biologic therapies or anti-inflammatory therapies for specifically depression. And the bottom part is something about the neural networks that Beata mentioned and how these are linked to inflammation that you can measure in the periphery. So you can see specific neural network abnormalities linked to peripheral blood measures of inflammation. So how does peripheral inflammation communicate with the brain? Well, we've been aware of that for a while, but there are two main pathways. One is the humoral or cellular pathway, either through active transport through the blood-brain barrier. There are in fact active transporters for things like TNF, direct entry through the circumventricular organs, or exogenous recruitment of immune cells into the brain. I'll say something about that later. The neural route, which is much more rapid, is essentially around vagal nerve detection of circulating cytokines and therefore afferent transmission to the CNS. And we can measure this both in the rodent using things like optogenetics and various forms of spatial transcriptomics, which I'll say something about in a minute, and in humans essentially around neuroimaging, both in terms of resting state MRI, magnetic resonance spectroscopy, which allows you to look at glutamate, for example, and molecular imaging such as SPECT and PET. So the two models I was talking about from preclinical studies, one is this so-called Aldara model. So this is a imiquimod, which is a skin-based treatment. So you shave the dorsum of the mouse and apply this to the back of the mouse, and it creates essentially an inflammatory skin disorder. So it's actually a model of psoriasis, another disorder which in fact has a huge amount of comorbid depression. So TLR7 agonist, and therefore the imiquimod binds to that, creates a peripheral immune response, and you get raised in interferon, cytokines, and chemokines in the peripheral blood. Okay. What's interesting is that we saw changes in the brain at a whole variety of different time points in a time series study. And these are at the transcriptional level, so we saw initially chemokine upregulation, not too surprising there in the sense that you get things like CXCL10, which is an antiviral chemokine, followed by the classical inflammatory cytokine responses, interferon, TNF, IL-6, and later IL-1 beta. So this is a series of studies we've done over many years now using this particular model in black sex mice and in a series of time series models. But that's transcriptional change. This also obtains at the level of the protein. Finicky to do in the brain, but we've been doing this with increasing levels of success actually. And what you see is exactly the same levels of, exactly the same molecules rather, at the protein level have been increased in the brain as a result of this peripheral skin-based distal stimulation. And what's intriguing about it is in fact the temporally distinct nature of it. In other words, the peripheral response will attenuate, which is what you'd expect, but the brain response is maintained. So there's almost, as it were, a slow burn in the brain, which is not resolving, and slightly worries me actually, because I'm thinking to myself, how does the brain resolve this? It's a very odd environment for inflammatory proteins to be present. It's an alien environment for immune cells to enter, and I'm not sure how the resolution happens, because this is not encephalitis. This is just an inflammatory response in the brain. The other thing we observe, and we've observed this several times now, so these are several replicated studies I'm summarizing here, is the ingress of exogenous immune cells into the brain, monocytes, natural killer cells, T cells. Now, you'll all be aware here in this audience that the endogenous immune cells of the brain are, of course, microglia, which are essentially macrophages, which has a unique cell lineage from the yolk sac, doesn't go anywhere else, very unusual. These cells are not supposed to be in the brain. Okay, they're certainly not supposed to be in brain parenchyma. And when I look down the microscope and be in my happy place with my microscope away from everybody else, I see these cells all the way throughout the brain parenchyma. And it's not what you expect to see. Okay, so it's not just perivascular. You're not just seeing these being extravasated through local vasculature. But I'll tell you about a technique we're going to try and use to try and solve some of this. So one of the puzzles we have is that we also saw, now is there any neural damage, I hear you ask? Well, certainly when you look at things like caspase, you don't see much in the way of, this is not killing neurons as such, but it is causing this strange kind of cloud phenomenon, which we can't quite explain. It's IgG, so it's an antibody. And they're surrounding neurons in these kind of wonderful kind of puffy clouds. And we're wondering whether this is actually the beginnings of damage. So you've got this almost autoimmune response at the neural level, but not actually killing the neuron. And it's in a variety of different areas of the mouse brain. You can see their cortex, particularly some subcortical structures, but not hippocampus, it seems. And I have a theory about the hippocampus being slightly protected from some of this, because the hippocampus is a whole series of atypical chemokine receptors expressed around its border, which the theory being that this stops any unnecessary cytokines and chemokines entering the hippocampus, but we haven't proved that yet, so it's anybody's guess. But in summary, this inflammatory stimulus, as imicamod, causes a small degree of blood-brain barrier leakage. We get astrocytic upregulation, microglial upregulation, so simple measures like GFAP and IBA1 staining, monocyte ingress. They're predominantly monocytic, these cells, I have to say. Cytokine and chemokine upregulation. And then we've just started looking at the protein level here in the third panel, a whole variety of different proteins we've been looking at, notably synaptic proteins, but also glutamatergic and antibody-based proteins, and also P2X7 related to the inflammasome, okay, in the microglia. So that's, the purine receptor that, will bind ATP, upregulate the inflammasome and microglia and produce IL-1 beta, which is, of course, pro-inflammatory. And so we think there are changes in synaptic plasticity, but not necessarily neural damage. And now we're starting to look at sickness behaviours. We've done this at a very basic level, looking at things like burrowing at the moment, but we're expanding that. And I'll maybe say something about that if we've got time at the end. So the second model is the maternal immune activation model. Why bother looking at this? Well, there is a long and rather distinguished history of an awareness that women who are pregnant, who are exposed to infection, trauma, stress, etc., have a less than optimal pregnancy. And that pregnancy seems to be related, that abnormality in the pregnancy seems to be related to upregulation in inflammation. And that's certainly been shown at the animal level, but also there are large epidemiological studies that have shown that women who've experienced that kind of stress, infection particularly, are at greater risk of difficulties with pregnancy, loss of the baby, and then also, intriguingly, those children who've experienced stress at an early life, so early life stress itself, so postpartum, they are at greater risk of developing inflammatory disease later in life. The Dunedin cohort, most famously, I think, showed that. And that is an intriguing, but as yet mechanistically unclear and rather opaque finding. But maternal immune activation has been something that's been linked to, for want of a better expression, psychotic-like behaviour. Okay, and here we infect the dam with usually LPS or a similar stimulus. In this case, we used resiquimod, which is a kind of relation of the TLR7 agonist I was telling you about. And J. Kwon, who did this work, looked at maternal brain, blood, spleen, and placenta. Very finicky, difficult work looking at mouse embryo brains, I have to say, but she did it, and found, very interestingly, upregulation of similar cytokines and chemokines in the embryonic brain. So this is after, normal pregnancy in the mouse, but an infected mum, and you get this whole variety of pro-inflammatory cytokines and chemokines upregulated. So she's still working on this, actually. Some of these chemokines are important at the bottom panel there because those chemokines are central to the recruitment of the immune cells I was telling you about. Okay, because chemokines, in case no one's familiar with them, are essentially small molecule cytokines that are essential both in neurodevelopment, so they're key to neurodevelopment, but also in terms of cell trafficking. Their role in neural development is in neural patterning, in essence, so ensuring that precursor neurons get to where they're supposed to go. If you knock out certain key chemokine receptors, for example, CXCR4, you will not develop a cerebellum, so they're pretty crucial. But in normal adult life, they're there to traffic immune cells to where they're supposed to go and deal with stress and infection. So all of this relates to systemic inflammation, but can we be more molecule specific? And to that end, we are trying to develop a new mouse model, which is an inducible TNF-driven model of inflammation. So these will be cell-specific producers of TNF, this key canonical cytokine in the brain. So we have developed a transgenic mouse to do this with a variety of different promoters. So P2R12, RY12 rather, for microglia, CD11B for monocytes, and the last one is allegedly for brain-specific endothelial cells. So this is with a colleague in Germany. I'm not 100% convinced by this yet, but we shall see, because obviously endothelial cells are everywhere, but if you can get brain-specific endothelial cells, and they are very much the Cinderella cell in this story, then that would be great. So this will allow us to overexpress TNF at specific points in time via particular cell types. So on the subject of being cell-specific, this is tricky, this area, because at the moment we've been looking at brain mush. Okay, so we're just in taking the brain, mushing it, and looking at, transcript and protein. But what we'd really like to do is look at the whole brain landscape from a transcriptional perspective, identify the regions that are relevant in terms of this upregulation, and then do a whole genome analysis against that specific region. So many of you might be aware of this new technology, Geomix, which allows you to do that. And it's now got something, there's now a child of Geomix, which is Cosmix apparently, which allows you to not only work out exactly what is upregulated where in a particular tissue, it's particularly used in cancer, but the next area is the brain, but also will allow you to tell which cells show a particular transcriptional upregulation. So it's very exciting, and we're about to try it and spectacularly fail, as you always do with these things, but we shall see. The other aspect is connectivity, and we touched a bit about that. And one of the techniques that we've just started using actually, and Rona McGonigal, one of our postdocs, has just got it to work, is Foxtrap. So this is exploiting the fact that CFOS is an early expressed gene. Activated neurons will express this at a snapshot in time very rapidly, usually within an hour and a half or so, and you can capture that using this technique. It's essentially cream ice that you can buy in and cross with whatever model you're using, and allows you to capture whatever neuronal ensemble is activated with whatever stimulus you're talking about. So for us, this is absolutely brilliant. So allows us to say, well, this neuronal ensemble seems to be important with, say, our imipramode model or whatever it happens to be. That will then allow us to target our optogenetics. Okay, so we can then say, well, which group of neurons do we actually want to see as linked to another group of neurons? And so that's what we're trying next. And then we can get our EFIS readout. And the last component is to use chemogenetic techniques where once we've identified that, then we can either excite or inhibit those relevant neurons and look at behavior. So that's the plan. And we've just started doing a bit of that. And then the last thing is really the translational element, which is to use some of the compounds I was telling you about at the beginning, namely these very specific cytokine inhibitors, and test those in the environment I've just described. The behavior is if we've got time, we'll say something about that, but it's a tricky business. So last thing is the human study, because I do occasionally see humans. And this is an experimental medicine studies we've just started, which to the MRC has this kind of experimental medicine portfolio where they don't really like anything to do with mice in this group. So you have to use it in humans, and you have to have something that makes a difference and it shows a mechanism. So here, we want to take patients with rheumatoid arthritis who express some of these psychiatric phenotypes I was telling you about, particularly depression. And we want to look at, so these patients are eligible for TNF antagonism, so that's a specific cytokine that antagonizes, and it makes a huge difference to patients. But what we want to look at is connectivity in the brain, yes, resting state connectivity. We also want to measure glutamate in the brain using, so this is high field strength MRI, so 7T, so it allows us to resolve the glutamate signal in a particular brain region. In this case, we're going to look at the insula. Why glutamate? Well, essentially, inflammation will upregulate glutaminase and downregulate glutamate transporters. So in essence, you get a net level of increased extrasynaptic glutamate, which we predict is excitotoxic. It might not be, but that's what we think is going on. It's a hypothesis. The last bit, which is the kind of experimental bit, which we will probably catastrophically fail at, but this is essentially taking the patient's own monocytes, sorting the blood for CD14 monocytes, labeling them radioactively, re-injecting them into the patient, and then working out whether they get into the brain or not using SPECT imaging. Please feel free to have a hollow laugh, but we will have a go at this. And that is essentially the model of a series of different experimental medicine paradigms that we're using. This is just one example of those studies. So I'll just finish off. The postdocs include Julie Bourguignon, Deepika Sharma, Jadok, Mona and Lilia, all of whom have done most of this work, as well as the PhD students and our technicians, and these are our funders and collaborators. Thanks.